All right, here's our last section, chapter 3, section 3. We're going to talk about methods in nucleic acid biochemistry. This is super cool. I always say everything's cool. So all of the techniques we're going to talk about are based on so many different fields. Like you have to understand enzymology, you have to understand molecular biology, genetics, microbiology. We take advantage of systems that are already in place and we make them work for us, which is super, super cool. So one of the most useful tools in biochemistry is the ability to isolate a gene and then express the protein product of that gene in an exogenous system. So what do I mean when I say exogenous system? I mean, if it's a human gene and a human protein, I can put that in bacteria and I can make it express the human protein like we do with insulin, for example. And we do this using plasmids. And plasmids are circular pieces of DNA. But in order to understand how we do all that, we have to understand chemical physical properties of cell membranes. We have to understand how prokaryotes versus eukaryotes are organized and all this kind of stuff. So when we talk about plasmids, plasmids are self-replicating pieces of cellular DNA. So when the chromosome replicates, that's independent of a plasmid. A plasmid can replicate without a chromosome replicating. So the plasmids carry extra genetic information that's not in the chromosomal DNA. So it's not duplicated. And a lot of times what you'll see is that that plasmid contains something that gives the organism an extra advantage. And we see them in both prokaryote and eukaryotic organisms, and they contain their own origin of replication. And that's what allows them to replicate independently of the host. So like independently of, let's say, the bacteria. So we can clone the plasmid, we can conjugate it, we can attach it to something, we can transform it or transduce it, which means put it from one organism to another organism. And it's just important to understand that the plasmid usually confers some kind of survival benefit to the host. Otherwise, it would be a waste of energy to maintain, right? So let's look at this. So this is our example inside of bacteria, right? There are a couple of different ways that this can happen. The first one is conjugation. In conjugation, we have basically bacterial mating, where we have horizontal gene transfer. So we have two bacteria, right? And they create a little conduit where their membranes merge and they can send plasmids either direction, right? So they can transfer a copy. But the thing to remember is that the original cell that had the plasmid has a plasmid, makes a copy of it, and then sends the copy to the other cell. So now both of the cells are, can maintain the plasmid, right? You can have transformation, where you have either a DNA fragment or a plasmid that actually is kind of floating just out in the ether, right? And the bacterial cell can take it up and put it in. And even if it's if it's a plasmid, it can just maintain the plasmid. But if it's a fragment of DNA, it can actually incorporate it into a plasmid and maintain it that way. And you can also have transduction. Transduction is from a virus. So you have a virus that has a piece of DNA, and that virus infects the bacteria and injects that DNA into the bacteria. And so usually what's on the vector or the plasmid is an antibiotic resistance. That's that survival benefit to having the plasmid. And so we can capitalize on this. We can use these as a tool. So here are some example plasmids that are used as cloning vectors. And so what you have are circular pieces of DNA that have an origin of replication, right? And then typically they have at least one, maybe two antibiotic resistance genes. So if you have the TET-R gene, it makes you resistant to tetracycline. If you have the ampicillin, AMP-R, that's what the R is for, makes you resistant to ampicillin. So bacteria that have this plasmid can grow on media that contain ampicillin, and they can grow on media that contain tetracycline. That would normally kill most bacteria. And so what we have is this place on this vector where we can insert DNA. And we can insert DNA due to restriction sites. So here's another one that has what are called restriction sites. And so if we have multiple restriction sites, we call it the MCS, or the multiple cloning, L-O-N, cloning site, right? And this is where we can insert DNA. So if we cut this plasmid with a restriction enzyme, an enzyme that cleaves the DNA at particular sequences, if we cut the plasmid with that enzyme and we cut a foreign DNA fragment, like the gene that codes for insulin, right? We can put that fragment that's been cut into the plasmid and we can ligate the ends together. We can glue the ends together. And now it's a new vector that has everything it originally had plus our DNA insert. And so once it has that DNA insert, we call this recombinant DNA, right? DNA insertion has occurred. So how do we check that that's happened? Well, we take this new plasmid and we put it back into bacteria. And we look for bacteria that are resistant to antibiotics. And sometimes you also have a vector that will have a gene like the LAC-Z gene. The LAC-Z gene, basically half of it, I can erase this, half of it is on one side of the multiple cloning site, and the other half is on the other side of the multiple cloning site. So if you stick a big chunk of DNA in there, it basically separates the two pieces of the LAC-Z gene and it no longer functions. So if you have no insert, you have a functional LAC-Z gene. If you have an insert, you have a non-functional. And so if LAC-Z is being transcribed, it's able to grow on media that contains what's called a substrate called X-gal. And the X-gal will be cleaved by the beta galactosidase gene, which is LAC-Z, right? And we'll have blue colonies because you'll be able to convert that X-gal substrate. If the LAC-Z gene is interrupted, then the enzyme's non-functional and it can't convert the X-gal into blue and our colonies will be white. So we call that a blue-white screening, or you can just do antibiotic resistance. I've done both, both, right? So we need to talk about those restriction enzymes that we use to cut the plasmid and we use to cut the DNA. And there are three kinds: type 1, type 2, and type 3. Type 2 is what we use in cloning. Type 1 and type 2 both require enzymes, and they don't actually cleave at specific recognition sites. So like type 1, if you have this site, the enzyme is going to cut 1,000 base pairs away from where you want it to. It's not specific. The type 3 is similar. It's a little bit more specific. It's like 24 to 26 base pair away from the recognition site. But what's really awesome about type 2 is that they cleave right at the restriction site, at the recognition site, right? And so let me show you an example. Here, for example, right, is EchoR1. EchoR1 is a type 2 restriction site. So there's a specific set of DNA bases that have to be present for the enzyme to recognize the DNA. In this case, it's CAATTC. So right here, CAATTC. EchoR1 will recognize that, and it's called an endonuclease because it cleaves inside of the recognition sequence. So that recognition sequence gets cut, right? And it basically gives half the recognition site to one side of the DNA and half of it to the other side of the DNA, right? And so this is really great because you're cleaving exactly the recognition sequence. You know where it's going to be cut. Now I can cut my foreign piece of DNA and stick it right in there, and I know exactly where it goes, right? So there are some that cause overhangs. This is an overhang right here. This part's the overhang. Here's a 2 base pair overhang, right? There are some that have 3' overhangs. So here are 3' overhangs. And there are some that have blunt ends. So that actually just cuts straight down the middle, right? So depending on your specific need, you're going to pick the right one. But they have all kinds of really interesting names. Hold on. What's interesting is how these restriction enzymes, these endonucleases, were named. They're named by the bacterial strain it was isolated from. So I don't think the one that I used as an example, there's one called Hindi 3, right? And the Hindi 3 is isolated from Haemophilus influenza. So Haemophilus, H-I-N. Look, I can't even spell an abbreviation, H-I-N-D. Haemophilus influenza, and it's strain D, and it's the 3rd enzyme that was isolated from this one strain. And so that's how they name them. That's why they're Echo R1, HPA2, PST1, right? That's where all these names come from. They actually do mean something, right? And the original use for these endonucleases is really, really cool. And they weren't designed for us to use, obviously, right? But we're hijacking them. So bacteria use these, and they evolved as a mechanism for the bacteria to fight against bacteriophages, so viruses that infect bacteria. And the way it works is bacterial DNA is replicated, and the new bacterial DNA gets methylated. And that methylation protects the DNA from these restriction enzymes. The restriction enzymes don't cleave methylated DNA. So when a virus comes along, a bacteriophage comes along and injects its DNA into the bacterial cell, what's going to happen is those restriction enzymes are going to see unmethylated DNA and go, oh, chew it up, cut it up, right? So that the bacteriophage can't replicate. But we've said, oh, this is a really cool mechanism. We can make this work for us. And it's really cool because these recognize specific DNA sequences, and we can capitalize on that, right? Okay, so how do we use restriction enzymes? If I can make it go to the next slide, that'd be great. All right, so we have our DNA, right? We have our vector. Here's our vector, right? And so we've isolated our insertion site, and that we're going to use Echo R1 as our restriction enzyme, right? And so we have not only our vector, but we also have our DNA of interest. And so what we have to find is a restriction enzyme that's on either side of our gene of interest and inside of our vector. And so a lot of vectors come with multiple restriction sites so that you can pick whatever works for you, right? And so we cut the vector with, let's say in this case, Echo R1. We cut the gene of interest with Echo R1. So we cut all of this junk goes away, and all we have left is our gene of interest. And so we treat both of them with our endonuclease, right? So now we have a cut site inside of our vector, and we have this important piece right here that we want. And so what happens is that we combine the two, and we use ligase to seal these gaps. Now we have a completed vector that has our gene of interest inside of it. We take that completed vector that has our gene of interest and we transform it into bacteria. So just like we said, bacteria can pick up those plasmids just from the environment, right? And then we let them grow. I mean, we let them grow on media that contains antibiotic, right? On the vector, we have the antibiotic resistance gene for, and you can also do the XGAL screening that I explained before, right? And so what we're looking for are colonies that will grow on the appropriate antibiotic. And if you're looking for the blue-white screening, you're looking for white colonies. So that's how we get the gene we want, a human gene, into a bacteria. And now we can tell that bacteria, go make insulin, right? or do whatever it is. I use that as my example, but there are so many other things that you can do. All right, let's talk about another technology, cDNA libraries. When you have cDNA libraries, now you're trying to do something a little bit different, right? In this case, we're starting with RNA as our starting material. Okay, and so what we do is we take mRNA pieces and we generate the complement of them in the DNA form. And so we use an enzyme called reverse transcriptase. And we actually found that in viruses that contain RNA as their primary genetic material, which then have to convert it to DNA before they infect a cell. Well, when they infect a cell, actually. And so we have this RNA, right? And we make new cDNA. And then what happens is okay, let's see. So we have our mRNA. We convert it to cDNA, complementary DNA. And then we use what's called ribonuclease to go in and basically cut up the RNA, because really what we want in the end is DNA because it's more stable. And so we have a complementary strand, and then we only, after this ribonuclease goes in, we only have little fragments of RNA left, and that's these orange pieces right here. We have fragments of RNA, and those fragments of RNA act like primers, and those primers can then be used to create the original strand of DNA, right? So we can then we have basically one complementary strand, and then we have pieces of DNA, and then we have to go in and use DNA ligase to ligate those gaps. And now we can take what was originally just an mRNA transcript and is now a double-stranded DNA piece and insert that into our vector. And once we insert that into our vector, now we have this library, because you're not only going to do this for one RNA transcript, you're going to do it for all the RNA transcripts in an entire cell, right? Or in one set of conditions. And now you have this library of all actively transcribed genes in one cell. And so each new bacterial colony will have a different sequence inside of its plasmid, and that sequence is specific to the mRNAs that are transcribed inside of the cell. So it's a really interesting method that allows you to gain a lot of information. So how do you know what that sequence is? Like, what if you don't know what that mRNA was and you want to sequence it? How do you sequence the DNA? And this is true. I'm going to explain DNA sequencing. So you can use DNA sequencing in what I was talking about with our cDNA libraries, or you can use it for anything else you can think of, right? So let me explain how sequencing works. And this is so cool because it was developed in 1977. This is amazing. And so the whole pivotal points of DNA sequencing is that you have to use dideoxynucleoside triphosphates. And what happens with those is these, if you insert them, if you have whatever sequence, I'm just going to use X's, right? A, T, T, C, C, G, whatever. When you insert this DD NTP, this dideoxynucleoside, what's going to happen is you can't add anything else after it. It won't work. And so the generation of this peptide, of this, not peptide, but of this DNA strand stops. And so it generates chain termination DNA molecules. So the DNA polymerase cannot add on another nucleotide, and so it stops. And so in addition to having it stop, it's also fluorescently tagged. It has a tag so that we can see it, And then we put it through a capillary gel electrophoresis that separates it by size, and we can detect it. So I'm going to show you how it all works. And this was, they got the Nobel Prize in 1980. There was actually two different ways, two different groups who designed two different methods, and they shared the Nobel Prize, and we've just basically used the Sanger sequencing method more and developed it more, and it's just more efficient. Okay, so one thing to kind of remember when we say, I know I said this I said NTPs. What is an NTP? It's any nucleoside triphosphate. So adenine triphosphate, right? Guanine triphosphate. It's any nucleotide, okay? So what do you start out with when you're doing sequencing? You're going to start out with a single-stranded, I am way too big, right? Okay, so I want single-stranded template DNA, right? And if you have double-stranded, you know that you can melt it and just increase the temperature, and you'll separate them, right? So you need a template, and you need a primer specific to that template. You need a DNA polymerase. You need your normal dNTPs. So this is what you would normally find in the cell. And you're going to have all of them: ATP, CTP, TTP, and GTP. Then you have these terminal nucleotides, the dideoxynucleosides, right, that are going to stop synthesis of the DNA strand when they get incorporated. But in addition to that, they also have a unique fluorescent chemical tag. So that's why they've colored them differently, because each one has a tag, but it's a different tag. It fluoresces a different color. And so that's why we have them that way, okay? So what's really important is that you have about a 300-to-1 ratio of dNTPs to ddNTPs, which means that you have a ton of nucleotides that can be put in, right? But you only have a little bit of the ones that are going to end DNA synthesis, right? And so when you do this, you're going to mix them all together, right? And what's going to happen is you're going to generate DNA products where the very end is always going to be a fluorescently labeled ddNTP, right? So everybody's going to start at the same place, and they're going to incorporate nucleotides until they incorporate one of those weird nucleotides, and then synthesis is going to stop. But at the same time, there's another strand that incorporated everything, one more, and then another fluorescent tag, right? And it goes on and on and on and on and on. And so what you end up with, right, when you do this is you end up with a range of sizes, right? And they're going to vary by one nucleotide. So you're going to have a bunch that are only only 2 nucleotides long, then a bunch that are 3 nucleotides long, a bunch that are 4 nucleotides long, right? Because that polymerase can't tell the difference between your dNTPs and your ddNTPs, it's going to incorporate them just as easily into the growing DNA strand. And so when you look at the products, right, you can separate them based on size. And so you look at the first one. The first one's going to be really small, and we're going to be able to see what fluorescent tag is at the end of that. That's only going to be 1, and it's going to be 1 fluorescent tag, right? So we're going to know that that first one is, for example, a C, because it's fluorescing in a certain color. Then we look at the one that is the next length, and we see that it's green. Well, that must be a guanine. That's a G. We look at the next length. That's an A. And so on and so on and so on. And that's how we sequence it. And you can sequence fragments this way that are about a maximum of 500 bases long. But you really need a lot of template DNA to do this. So when you do this, before you can sequence your DNA, usually what you have to do is amplify your DNA. So how do you amplify your DNA? You're going to amplify your DNA with PCR. So this is a process that amplifies a specific sequence of DNA. And so it's really replication replication reaction repeated in many cycles. Many cycles. Okay, so it's just something that cycles over and over and over again, right? And so our number of DNA molecules will increase 2 raised to the n in each cycle. Okay, so for example, if we do a cycle that's 20 cycles long, right? So you do 2 raised to the 20. If you put that into your calculator from one template, so if you had one template, one piece of DNA, at the end of this, you would end up with about 1 million copies of that one template, right? And each cycle only takes a few minutes. So in a few hours, you can amplify a single gene very, very efficiently. And it does depend on the gene length. The longer the gene, the longer the cycle. But so you do this in 3 phases. And remember that we can denature DNA just by temperature. So what do you first want to do is you're starting with basically double-stranded DNA, right? So what do you want to do in the first stage? I want to separate it. I want to go from double-stranded DNA to single-stranded DNA. So that's my denaturation. I'm separating them, right? Phase 2 is annealing. So I have my single-stranded DNA and I want to anneal a primer to that single-stranded DNA. And then in phase 3, okay, there's my primer, here's my original strand, In phase 3, I want to now extend from the primer and make a complete copy of those DNA molecules. So those are my 3 phases. So if we look at this kind of in a graphical format, right? And those 3 steps are going to occur in about 2 minutes, which is really kind of cool. So what are you going to start with? You start with your target double-stranded DNA sequence. This is double-stranded DNA, right? You have primers. You have primers that are going to anneal. One's going to anneal here and the other's going to anneal over here, right? Those are the target sites for your primers. And then you also need polymerase, but you can't just have any old polymerase. You need to have Taq polymerase. Taq polymerase is isolated from a heat-stable organism, T. aquaticus, and it functions at very high temperatures. If you use regular polymerase, it's going to be inactive at the temperatures that are required for PCR. So the discovery of Taq polymerase was facilitated PCR from happening. Another one of our examples of us hijacking a normal system. I just love that we do that. Okay. And then you also need, I guess they don't really show it, but you do need DNTPs. They didn't put it. That's terrible. You need DNTPs, right? So you have to have a target sequence. You need 4 things. You need a target sequence. You need a primer, right? Well, you really need 2 primers, but primers. You need DNA polymerase and you need DNTPs. Okay, now we have everything we want. So now 3 phases, right? Phase 1, I'm going to do phase 1, PS1. We're going to denature the DNA, right, with high temperature. So we're going to separate it into single-stranded DNA. So now we have single-stranded DNA. Then we're going to cool it. So this is phase 2. We're going to cool it to promote the primer annealing to our single-stranded DNA. And then in phase 3, we're going to extend the DNA with the Taq polymerase. Now, we have 2 copies of our target DNA. Well, guess what we're going to do? Again, we're going to denature. There's phase 1. We're going to cool it to promote primary annealing, phase 2, and then we're going to extend with Taq polymerase, phase 3. What do we end up with? 4 copies. So this is why you're going to get so many more, because every time you go through a cycle, you're going to duplicate however much DNA you had there. So if you went for 30 cycles, 30 cycles, so that's 2 raised to the 30, that gives you about a billion copies. It's amazing, right? Amazing. And it's really great because you really only had to start with one piece of DNA. It's kind of cool. You could use not only that, like it could be something you isolate in the lab, but it also could be from a patient sample. So maybe from a cheek swab or something like that, where you don't really normally get a lot of DNA from that sample. Okay, so we went over this. Yeah, we did all that, right? Okay, we did that. All right. So sometimes you also want to be able to look at the level of gene expression, not just, is this gene being expressed, right? We talked about how to do that. We can take our mRNA transcripts, turn them into cDNA, put them in the vector, sequence it, and say, yes, okay, this gene was transcribed. But sometimes we want to say, well, we know this gene is transcribed, but what happens when we change the conditions? Does it go up? Do we transcribe more of that gene? Do we transcribe less of that gene? So there are two different ways that we can do this. We can look at gene expression microarrays, or we can use RNA sequencing. So this, we call this RNA-seq. That's our kind of abbreviation for it. And there are two different ways, but they give you basically the same information out. And so we're going to go over each one and how they're comparable, basically. So when we talk about gene expression microarray, the most commonly used one is called a gene chip. And so what you have, right, is this little bitty chip. And you take DNA sequences and you put 6 million DNA locations per chip. Now be careful with that. A DNA location is like one gene. Okay, so you have more than 6,000 genes on the chip, but at each location on the chip, right, you have about 1 million of each unique sequence, right? So what that means is you have one gene, right? And a gene can be really, really, really long. And so what happens is they take pieces of that gene and they attach pieces of that gene to one location on the chip. And you have multiple copies of each of those pieces. And so this quadrant right here on the chip corresponds to gene X, right? And so you have all these different pieces of gene X and multiple copies of each of the different pieces, right? So you have millions of identical single-stranded DNA strands at each location. And you take some set of cells here, right? Let's say maybe you have normal cells and you want to do the same thing. And so you're going to have another container of cells, right? And this is going to be cancer cells. And so I want to compare what's my mRNA changes when I have cancer, right? So I'm going to do the same pathway for my cancer cells that I do for my normal cells. I'm going to isolate my mRNA. I'm going to use a primer that anneals to my poly A tail, because remember, this happens to all of our mRNA transcripts. They get that poly A tail. So you take a poly T primer and anneal it, right? And then you start to make these unique single-stranded cDNA strands, right? And then you put in DNA polymerase and ribonuclease H, just like we did before, and it cuts up, right? It cuts up the mRNA, and little pieces of the mRNA are left, and so then you can make the complementary DNA, right? And so you're left with double-stranded, and we call this cDNA. So anytime you start from mRNA and go to DNA, so you go backwards, we call that cDNA because it's complementary DNA. Then what we do is we take those and we label these pieces. We label them with a fluorescent probe. And once we have those, we mix them with this chip. So we put our fluorescently labeled cDNA probes on this chip, and we basically rinse it, right? And then we see what attached, because anywhere where it attached, we have probes. So here, so this is all of our cDNA that has that fluorescent probe on it. And so anywhere where it's green, we know it's fluorescing. And the intensity of the green will tell us how much it's fluorescing, or how much of the cDNA fluorescently tagged fragments have attached to the chip. So the more mRNA was there, the more fluorescent that little piece of the array chip is going to be. And so then you can compare two different conditions and say, you know, here's I have two chips, right? Here's my normal, here's my cancer. And what I can see is the quadrants, right? Each of these quadrants represents something else. In a normal cell, I have high levels of these sets of proteins, but in cancer cells, I have a different set of proteins or mRNA transcripts that are present, right? And so we can see changes between two different conditions. And there's an animation. Oh yeah, there's an animation I wanted to show y'all. Let me see. Oh, hang on. You can go and I'm not going to play this for you, but you can go onto the Norton website. Will this come over? Oh, come on. Okay, wait, let me stop the share. And then I'm gonna try to share this. So let's see. If I bring this over, right? And I'm gonna share again. Desktop one, share. Okay, I think I'm sharing. What you're gonna see is if you go to your Norton website, right? And you go to animations. If you go in chapter 3, we have our first animation, and this is the gene expression chip analysis animation. It's pretty cool. You should do it. You should absolutely do it. Okay, let me make this bigger again. All right. So that's the gene chip technology, and that's the animation you can go to in your book. Then the other way to do this is to do next generation sequencing analysis, right? So it's the same thing. You start with your cultured cells and you isolate your mRNA. Now that you have your mRNA, you convert that to cDNA. So this is pretty similar, pretty standard as to what we did before, right? And then what we do is we PCR amplify our cDNA, right? We went mRNA, cDNA, and then we amplified it. When we amplify it, we then sequence this. So just like we talked about before with the Sanger sequencing, we actually look at the individual residues and do high throughput sequencing. And basically you get very, very similar results. But the difference is that there's no bias when you do this next generation sequencing or the RNA. This is RNA sequencing. You're not actually sequencing RNA. You're actually going from mRNA to cDNA, PCRing it, and then sequencing it. But there's no bias, right? In that chip, you had a predetermined set of genes that were already on the chip. So maybe you didn't put a gene that you were supposed to put on there, right? And so it allows you to not have a bias. Also with RNA sequencing, you can have a low abundance of something, like 5 copies of a transcript, and you'll still see it, but you might not see it with the chip. You can also see alternatively spliced transcripts, right? Remember we talked about before where you had, you know, exons 1 through 6, and this one got exons 1 through 4, this one got exons 1, 3, 4, 5, and 6. And so you can actually see those alternatively spliced transcripts with RNA sequencing, but it's more expensive and it's more time consuming. So, you know, it depends. But if you're just looking for, I know my gene, is it upregulated or downregulated? It will tell you. It will absolutely tell you. Because, and I'll just look at the end data, right? We have 2 genes, gene X, right? And we have two different ways that we're assaying them, with the array and with the RNA sequencing. And so we have our treated versus our untreated. So if we look at gene X, what we can see with gene X is that when we go from treated to untreated, our number of gene copies went down, or you go from untreated to treated and it goes up, however you want to think about it. But if we do the same comparison with our RNA sequencing, so same gene, we're starting with, and it doesn't really matter how many copies you have. All you're looking for is it upregulated? Is it downregulated? And here we also see a downregulation. So it gives you the same results. All right, and I think that is the end. Ooh, that is the end. All right, so that was the end of chapter 3, section 3. Gosh, I could teach on that for forever, but it was really fun. So I hope you had fun.